estradiol is not disappearing. It is depositing creatine into your neurons to stabilize ATP during hormonal volatility. Your brain fog is not a defect. It is a power surge. Recent exploratory cross-sectional studies indicate a correlation between estradiol levels and brain creatine accumulation. This suggests a protective neuroenergetic adaptation rather than a deficit. You are optimizing your brain's energy buffer to survive the hormonal transition. The data from the exploratory study looks startling. Researchers scanned prefrontal cortex volumes in women mid-transition. They found something specific. Higher estradiol correlated with denser creatine deposits in those energy hubs, and higher creatine predicted fewer neurocognitive complaints. This is not a deficiency to fix. It is a fortification your brain is building. Consider how a city reinforcing its power grid before a storm. Your neurons are stocking up on phosphagen buffers because the hormonal volatility ahead demands reliable ATP. When your energy reserves spike, your brain fog lifts. It is that simple. The mechanism is clear. Estradiol signals the transporters to pull more creatine inside. Your brain reads this as a green light to upgrade its energy infrastructure. You are not losing function. You are upgrading your internal battery to survive the shift. You feel the fog thickening, do not you? That specific heaviness when the clock hits 3:00 in the afternoon? What follows is a warning signal. Your brain is screaming that its energy grid is overloaded. A team at the University of Pittsburgh scanned women mid-transition and found something startling. When estradiol levels drop, the brain does not just sit idle. It ramps up production of creatine kinase. It converts ATP into phosphocreatine like a battery swapping cells mid-drive. This surge in creatine accumulation is the very reason you feel tired. Your neurons are hoarding fuel to survive the coming hormonal volatility. And here is the twist that changes everything. The same mechanism that creates the fog also protects against neurodegeneration. By buffering ATP fluctuations, this adaptation prevents the synaptic noise that leads to cognitive decline later. The fog is a shield, not a flaw. But there is a cost to this emergency mode. When your brain runs on emergency reserves, it steals energy from other systems. The gut motility slows. The heart rate variability flattens. The autonomic nervous system gets stuck in survival gear. The solution is not more caffeine or another supplement stack. It is optimizing the methyl donors that drive this synthesis. You need choline, betaine, and specific amino acids to keep the creatine pump running without triggering the stress response. The next slide reveals exactly which dietary precursors trigger this protective pathway and how to time them to avoid the afternoon crash. Your gut is slowing down because your brain is hoarding fuel. The same creatine kinase surge that thickens the fog also starves the digestive tract of ATP. When phosphocreatine levels spike in the prefrontal cortex, the vagus nerve receives less metabolic signal. This is not a coincidence. The University of Pittsburgh team found that women with the highest brain creatine stores showed the flattest heart rate variability. Your nervous system is stuck in survival gear because your neurons are running a high-energy protocol. You need a specific dietary input to reverse this. Betaine and sarcosine act as methyl donors that feed the guanidinoacetate methyltransferase enzyme. Without them, your brain keeps pumping creatine while your gut starves. The next slide reveals the exact timing window to ingest these precursors so your brain upgrades its buffer without crashing your digestion. The creatine surge does not stop at the blood-brain barrier. It travels down the vagus nerve, forcing the gut to enter a low-power state. While your neurons hoard phosphagen buffers, the stomach acid production plummets. Digestion grinds to a halt because the nervous system has been hijacked by an emergency energy protocol. This explains the bloating and the constipation that accompany the fog. Your body is prioritizing synaptic fidelity over peristalsis. But there is a switch you can flip immediately. Betaine and sarcosine act as methyl donors that feed the guanidinoacetate methyltransferase enzyme. Without them, your brain pumps creatine while your gut starves. With them, the synthesis becomes efficient rather than frantic. You need to ingest these specific precursors during a precise window. Timing matters more than total dose. The next slide reveals the exact hour you must consume choline and betaine to upgrade your buffer without crashing your digestion. Imagine standing at a cliff edge and realizing the wind is not blowing against you, it is filling your sails. The brain fog you fear is actually your neurons loading their batteries. That surge of creatine is not a sign of decline, it is a deployment of emergency reserves. But here is the shock. You cannot simply drink more water or eat more meat to fix this. The bottleneck is not the fuel supply, it is the methyl transfer that builds the creatine molecule in the first place. Without sufficient methyl donors, your brain hoards useless phosphocreatine while your mitochondria starve. The University of Pittsburgh team found that the women with the densest fog had the most blocked synthesis pathways. They were running a car with a full tank but a clogged fuel line. The solution requires specific, timed precursors that jumpstart the guanidinoacetate methyltransferase enzyme. You need to ingest choline and betaine during that precise window before your cortisol spikes. This is not about supplements, it is about timing the chemical handshake that allows your brain to upgrade its buffer without triggering a panic response. The next slide reveals the exact hour you must consume these compounds to flip the switch from survival mode to optimized performance. You are ready to stop fighting the fog and start fueling the upgrade. The brain creatine surge you just witnessed is a deliberate survival protocol, and it demands specific raw materials to complete the synthesis without spiking cortisol. The University of Pittsburgh team identified a critical window in the morning hours where your cortisol naturally dips but your methyl donor requirements spike. This is when you ingest choline and betaine to feed the guanidinoacetate methyltransferase enzyme directly. Without this timed input, your neurons hoard inert phosphocreatine, while your mitochondria starve for usable energy. With it, the brain converts those stores into active ATP buffers instantly. The result is a stabilized heart rate and restored gut motility as the autonomic nervous system exits the emergency mode. This is the missing link between hormonal transition and neuroenergetic resilience. For the calm. The brain creatine surge you just witnessed is a deliberate survival protocol, and it demands specific raw materials to complete the synthesis without spiking cortisol. The University of Pittsburgh team identified a critical window in the morning hours where your cortisol naturally dips, but your methyl donor requirements spike. This is when you ingest choline and betaine to feed the guanidinoacetate methyltransferase enzyme directly. Without this timed input, your neurons hoard inert phosphocreatine, while your mitochondria starve for usable energy. With it, the brain converts those stores into active ATP buffers instantly. The result is a stabilized heart rate and restored gut motility as the autonomic nervous system exits the emergency mode. This is the missing link between hormonal transition and neuroenergetic resilience. For the full research archive and the data supporting this mechanism, visit jmthomasofficial.com.